Not all drugs are effective in all patients. Pharmacogenomics gives us the opportunity to understand a patient's metabolism status before we prescribe a therapy. This reduces the trial and error that's often required to find an effective therapy. It reduces the time to benefit for a patient, and potentially it avoids adverse events. Now, my first experience with pharmacogenetics was in the late 1980s. Now, I'm not old enough to have been in the laboratory back then. I was a young boy with appendicitis. In fact, I had my appendix removed and I was prescribed a combination of Panadol and codeine as pain therapy. And I remember thinking back then, it didn't work. I was pressing that call buzzer like it was a Halloween doorbell, trying to get these nurses to come in. Was I a complaining young boy? As it turns out, no, I wasn't. I'm a poor metabolizer of CYP2D6, star 4 homozygote. And it took several years, actually 30 years, before I found out that result. I had been prescribed codeine on a number of occasions, and on none of those occasions it worked. Now, CYP2D6 is a complex gene. It's been amplified into homologs. Those homologs have then fused and formed non-functional hybrid genes. Today, Agena is launching our Veridose portfolio, a suite of panels that does genotyping of pharmacogenetics genes, but also does highly accurate and efficient typing of this complex CYP2D6, the copy number variations and the hybrids that occur. Now, I'm pleased to introduce to you today our two speakers, both experts in the field of pharmacogenomics, but having different aspects. Our first speaker, Dr. Houda Hachad, is going to talk to us about taking the typed alleles in the pharmacogenomics genes and translating those into the functional status or phenotype. Dr. Wachtler is going to talk to us from a laboratory's point of view, implementing technologies for pharmacogenomics characterization. So I'd like to ask Dr. Hachad to join us. Dr. Hachad is an entrepreneurial scientist with deep experience in pharmacology and pharmacogenetics. At the University of Washington, she co-developed two knowledge-based technologies, a drug interaction database, as well as a pharmacogenetics database. Houda is currently the Chief Scientific Officer at Translational Software. Agena Bioscience is pleased to be partnered with Translational Software with their Precision Health Nexus suite to accelerate the use of genomic data that comes from the Mass Array platform for smarter and more comprehensive healthcare solutions.
Thank you, Darryl. Hi, everybody, and thank you for having me today. It is really a pleasure to be here and to recognize some faces that are people who are trying to move the pharmacogenomic implementation forward. Thank you for being here, and thank you, Darryl, for this introduction. So the title of my talk, I don't know how to get these slides, maybe with this. Yeah.
No, it's backwards, the bottom one is.
Oh, this one. Nope. Some technical difficulties. So, so today, the purpose of this talk is really to talk about challenges when we are testing for CYP2D6 genetic variations, namely these hybrid structures, as Darryl was pointed out, that result from these fusions with neighboring pseudogenes. So I will be giving a brief introduction on the clinical utility of CYP2D6 genotyping, examples of complexities and solutions that exist as of today to help us move forward, as well as some case studies that illustrates really some of these issues. But before that, I just want to briefly tell you who we are. So we are a technology company. We're located in the Seattle area, and we've been offering services around pharmacogenomics since 2013. So we have a suite of software capabilities that allow multiple partners to establish their pharmacogenomic programs. And as you can see here, our Nexus approach is sort of a network of players or stakeholders in the pharmacogenomic implementation, namely diagnostic laboratories, healthcare organization, as well as other software vendors that might be utilizing our content or our technologies to overlay their solutions on the electronic medical records. We also work with some novel programs such as benefit managers that are interested in bringing the pharmacogenomic approach into optimizing therapies for patients. So, so far we have done millions of translations and interpretation of results since the past years, and we have reached thousands of physicians through our reporting and clinical decision support solutions. And as I was saying earlier, we have integrated with plethora of platforms that are either standalone or integrated in electronic medical systems. So why did we choose pharmacogenomics? So as you already know, precision medicine has multiple applications, but pharmacogenomics has been around for many years in the research area or during drug development. So we have excellent evidence that supports the genetic as a cause for drug response. We can also notice that a lot of people, more and more people are taking medication, and as a matter of fact, more people are taking multiple medications. So the impact there is very profound. And finally, we are lucky. We are one of the genetic applications that has a vibrant community of experts that was able to get together to constitute groups and experts groups and consortiums in order to produce evidence-based reviews and evidence practice guidelines for the doctor to be able to utilize that knowledge. And finally, as Darryl was pointing out, there is, with pharmacogenomic signature, you can do a perpetual use of that information over time. So if Darryl was lucky to be born these days, and we would have profiled him, we would have been able to detect this poor metabolism status for this gene, and we would have been able to prevent these unnecessary inefficacy or lack of response with codeine, for example. So the tools that are today helping us to implement pharmacogenomics are the availability of multiple platforms and technologies that are cost-effective and allow us to do with good turnaround to obtain results preemptively for the doctors before they prescribe these medications. We have these large databases that have been compiled over the years that constitute these repositories that allow us to standardize these annotations and these designations of these pharmacogenomic either variations as well as the associations with the drugs. And finally, we have now reference material that we can utilize that can help laboratories validate their assay and move forward and offer these tests. As I pointed out earlier, we have excellent evidence, and we also have in the FDA prescribing labels additional information that indicates that the agency is really concerned about having in a population X% of individuals that might be not responding or responding in a different way to medications. We have expert groups that are trying to help standardize the field, coming up with standards on what is the minimum of variations that need to be tested for a gene, standards to allow us to translate these genetic signatures into a likely phenotype that we can then utilize to select the medications and the recommendations that pertain to these phenotypes. So CYP2D6 is the ideal candidate for PGX implementation. It encodes a metabolizing enzyme. So it's these families of enzymes metabolize xenobiotics, not just medications, but they are also known for metabolizing endogenous substrates as well as toxins. And they're localized in different organs, mainly the liver, which is the main biodetoxification organ, but you can also find them in other tissues such as the intestine and brain. Within the CYP family, CYP2D6 is very interesting in terms of maturation trajectory, because we notice that right after birth, within the couple weeks after birth, the gene expression reaches full maturity within that month. So that gives us an opportunity to start utilizing and allowing us to utilize genetic information in really young children. Other genes, such as CYP2C9 or CYP2C19, the maturation trajectory is slightly different, and it takes a little bit longer before that gene expression is fully mature. So most interestingly, if we look at the world of the commonly prescribed medications for common illnesses, we realize that up to 25% of these commonly prescribed medications are metabolized to some extent by CYP2D6, which means that if we have varieties of phenotype, it is likely that CYP2D6 will be impacting different therapeutic areas. And we can see that from the number of evidence practice guidelines that have been issued by the Consortium for Pharmacogenomic Implementation. And you can see we have so far 6 guidelines that span different therapeutic categories and specialties. We have 2 guidelines that pertain to psychotropics. We have a guideline for a major anti-cancer medication, and we also have guidelines for therapies that are used in addition to anti-cancer therapies such as antiemetics. So the relevant, if we examine these practice guidelines, this is a list of common medications that you will be seeing in pharmacogenomic reports or that have been implemented preemptively in clinical organizations to guide treatment with the use of CYP2D6. Again, you can see that psychotropics constitute the larger family of medications that are impacted, but cardiology has also a number of medications. Some of them are commonly used, but some of them are rarely used, and these could be also affected dramatically by CYP2D6 phenotypes. So now moving on to the next portion of my talk, which speaks of complexities. One of the observations that we can say about CYP2D6 phenotypes is that if we examine the CYP2D6 activity among individuals, the traditional approach was using phenotyping. So that is before the era of genotyping. In the late 70s and 80s, we would give an individual a medication that we call a probe substrate, either spartine or the brizoquine or dextromethorphan, and then we would measure the metabolite in urine. And by doing a metabolic ratio of that metabolite to the parent, because we knew that metabolite was only metabolized by that enzyme, we can use that ratio to actually classify individuals into these 3 to 4 major groups: individuals that had very high activity, those that had almost no activity or low activity, and those that fall in between that we call a normal to intermediate. So this is sort of the common traditional way of presenting these phenotypes. And actually the terms metabolizer, a low metabolizer, slow metabolizer, or ultra rapid, was developed based on phenotypic observation, not on genotypic observation. Later on, when the gene was actually cloned and we were able to link genetic variations to these expressions of these phenotypes, we realized that if you can just use genotype and you can examine the type of alleles this individual was carrying and be able to find a correlation to some extent with the phenotype. And you can see it here on this slide where individuals that have genetically are, oops, sorry, I went too fast, genetically are predicted to be poor metabolizers in red. When we predict based on genotype, based on variations, there is a very, very good correlation with the expression, the phenotypic expression, when we measure it with this metabolic ratio. This is not necessarily true for the other phenotypes where you can see that there is overlap between certain phenotypes, which means that there is this, there are individuals that you can genotypically predict to be intermediate metabolizers or normal intermediate metabolizers, but you can, when you measure the phenotype, they turned out to be normal metabolizers. So this is something that we need to keep in mind, is when we do the genotype to phenotype prediction, it's a likely prediction. There are methods, of course, that have been refined over time to predict based on the function of these alleles you detect, how you can get to a likely phenotype more accurately. And one of the works that the recent standard, for those that follow the standardization from genotype to phenotype translation, looked at all the body of evidence to be able to refine these classifications. So we, based on these phenotype observations, the second challenge that we know about CYP2D6 phenotypes is their frequency. If you look at these major ethnic groups, some phenotypes such as the intermediate metabolizers, oops, I'm going too fast here, I think, are pretty equivalent across ethnicities. But when you look at ultra-rapid metabolizers, you realize quickly that some populations have extremely high prevalence of that phenotype trait. The same is true for poor metabolizer, but what is very interesting to note is every ethnic group has these expressions. Sorry, I think my slides are going too fast. Every major ethnic group has these 4 phenotype traits. So it is not true when we are running a test for a homogeneous ethnic group to say that we're more likely to be seeing this. We have to acknowledge that some of these individuals will have other extreme phenotypic expressions. Which brings me to the gene itself and its complexity. And as Darryl was pointing out, if you look at CYP2D6, it's located next to 2 neighboring pseudogenes, which adds additional complexity. It's rather a small gene, and there are some interesting features when you look at the gene and the neighboring pseudogene 2D7, is that they have this area here in blue, which is very common. It's a common and identical area in the downstream region of the gene. But there is a difference in that spacer. There is a sequence after CYP2D6 before you get to this repetitive element. And by the way, these repetitive elements in the downstream region are almost identical. So this presents challenges when you're designing your assays because of this high degree of homology. Oops. Okay. I don't know what's going on. Okay. So if we look at the type of genetic variations that we know about the gene, there are 2 types. There's the sequence variations, the SNPs and small indels, and then you have those that pose problem, which are the structural variations. And these could be copy number variations, where you have full gene deletion, duplications, multiplications, but you also have these complicated structures with these gene rearrangements with the neighboring pseudogene 2D7. And these could be hybrids as single entities, single gene units, but they can also be in tandem with a CYP2D6 allele. So as you can imagine, this presents additional challenges when you are trying to detect these extra copies of CYP2D6, because when we have extra copies, we might get into these ultra-metabolizers trait if the functional effect is there. So the entity that have taken over this cataloging task and this daunting task of documenting CYP2D6 alleles is Pharm.D. And Pharm.D, what we do with CYP2D6 is we receive these submissions of these alleles, structures, full submissions, we review them, we catalog, and we decide, is this a new allele, is this an existing allele for which we have a full sequence, is this a sub-allele, is this a major allele, but it's just a different flavor of it. So far we have definitions for over 139 core alleles, and we get submissions every month, and we review and add these variations as we see them. So Pharm.D has a wonderful website. I invite you to all become experts and use it. It has educational material, and please, if there are any questions or feedback, we would love to hear from you. So prevalence of these common alleles, as I said earlier, the phenotype frequencies are different, but the commonly tested alleles that actually are resulting in reduced activity or no activity, the prevalence could be also slightly different among ethnic groups. And what this slide is showing is the frequencies of the deletions and the duplications, which are some of the major structure variations that we look for into these assays, are also very prevalent, very different among ethnicities. As you can see here in Africans, you have to make sure that the assay is looking for duplications and can actually discriminate between the existence of a hybrid and versus a duplication. And we're going to get into some examples in a minute. So the duplication and multiplication events are described in this slide. So this is a small cartoon that you can find on Pharm.D. We have examples, and we explain all of these events. When we talk about duplications and multiplications, it's when we have identical alleles, two or more, that are inherited from the same parent and are on the same chromosome. And as you can see, the common ones are the star 2, the star 4 could have also multiplications. The star 1 allele, which is the reference allele, can have also duplications and multiplications. But we also need to know that we have the alleles that can be duplicated can be normal function, can be reduced function, but can be also no function, which means that the impact on the phenotype is going to be different. So proper characterization of the allele, as well as the count of this allele, is key in order for us to do a good prediction for that phenotype. So presence of multiple copies may or may not alter the function, and that is very important to keep in mind. So to do this translation from genotype or diplotype to phenotype, we need to properly examine the number of alleles that we have, but we also need to know their function. And as you can see here, this is sort of the continuum of phenotypes, that you can observe in a population, the poor, intermediate, and normal. And these are examples of diplotypes that can have either 2 gene units or multiple gene units, but they will all result into the same intermediate or the same phenotype group. So we notice that sometimes there is misunderstanding by some laboratories that when you have extra copies, it will result into the ultrametabolizer trait. This is something that really needs to be looked at carefully. And this illustrates it beautifully because you can have an individual who has 2 copies of a reduced function allele. I don't know what's going on with these slides, but it's still exhibiting a normal phenotype trait. An example would be this star 1, star 41, 2. Because when you have 2 copies of a reduced function, it does not necessarily result into an increase in the enzyme function. So this is something very important. The recent genotype to phenotype relationship that was proposed after surveys of experts is really coming up with this continuum and will help us tremendously into phenotype assignment. Now, moving on to the complexities, after duplications and deletions, the hybrids are the example of complexities and situations that can be challenging to interpret or to detect. So the first example are these 36 and 3610 tandem alleles, hybrid alleles. And as you can see from this cartoon here, the 36 allele is basically a star 10 allele, but it has, in the downstream region in exon 9, there is actually a small region, a small sequence here in red, that is a CYP2D6 sequence. So a portion of the 2D7 was converted and is actually has been embedded in this region. So the challenge with this particular allele is if you're running CNVs, copy number variation, that is in this exon 9 region, you can imagine that you're going to have some dropout. And the probe might look like it will not amplify, and you will be getting a lower number of copies, which might indicate a deletion, and as a matter of fact, it's not. So in order to do that, you need to be able to detect multiple regions, copy numbers in spanning multiple regions, and compare it to ensure that you actually do have a deletion, or you might have just a 36 down there. The problem here is that 36 exists also in tandem with the star 10. So there is a 10 here, and next to it you have a 36 with this rep 7, and that adds additional complexity into the detection based on CNVs. So bottom line, if you're using CNV by looking only at a specific region, and if you're picking the wrong region, you're very likely to be missing these particular structures. So the frequencies of these alleles, interestingly, the 36 phenomenon or the type of hybrids are very, very common in East Asians. And as you can see here, especially the 36, when it's in tandem with the 10, is very, very frequent. And as you can see, it's between 12 and 34%. So if you're doing assays, you will very likely be getting these discordant CNV results very frequently for these populations. The 36 by itself is less common, but studies in Chinese in Hong Kong have indicated up to 3%. So this is something very prevalent to keep in mind. It is very important to note that while the 3610 does not change the function, the 36 actually does because it's a no function. So interestingly enough, recently two independent groups, one of the authors actually is in the audience, have identified the 36 as being also prevalent in African American. So this is not an East Asian a trait, but you can also have it in African American, which can pose very problems in the interpretation. The next hybrid is the 68, and the 68 now, as you can see, the difference between the 68 and the 36 is the large portion of the 2D7 that was embedded in the 2D6. This is known to be existing in tandem with the 4, but also in a complicated structure with the 13. The 6-2D6, the recent data indicates that when you detect a 4, it's actually up to 20% of the 4s can have these 68 4 tandems. So this is something to keep in mind. It will not change the phenotype because both of them are no function, but it will create these discordant CNV results and make it difficult for interpretation. The 4N is a different tandem. It is different hybrid that is similar to the 36 because it has that similar exon 9 conversion from the 2D7, which you can see here in red. And it also can exist as a single entity or in tandem with another 4 or multiple copies of the 4. So up to 1.5% of the 4s are actually tandems or 4N. And while it will not change function, it will create this complexity when you do interpretation. So if we take the whole, all of the 36 issues, or other hybrids that actually can mimic the 10, you'll realize that a lot of platforms that cannot detect these hybrids will miss them, and they will all be collectively called a star 10. And that is a mischaracterization of a diplotype. So the final complicated hybrid to detect is a 13. The 13 this time is not in the downstream region of the gene of 2D6, but it's in the upstream region where the 2D7 structure is present, and then it will switch back to actually a 2D6. The ones for which we know we have some data about frequency, these are rare events, are these two where they can be in tandem with the star 2. And this can pose problems when you are detecting star 2 duplications. And we have an example of a case study that illustrates that. So this is sort of the structure that is known for the 2 hybrids, for the 13 with the 2. It's low prevalence, but as you can see, you can find it in 3 different ethnicities. So it is very important to keep in mind that when you're detecting a star 2 duplication, this actually might be just a hybrid with the 13, which doesn't change the function. And the issue here is that you might be mischaracterizing patients and calling them ultra-rapid metabolizers. As a matter of fact, they're normal metabolizers. So there is a beautiful paper in 2012 that gives you an idea of the prevalence of these structures, and if we do a proper calling of these structures, how you will be able to refine these phenotypes. This is summarized here, where over 20% of the 10s were actually tandems, and up to 1% had actually an undetected hybrid allele. So this is pretty consistent, pretty common, and up to 7% actually that were characterized as duplications were, a matter of fact, hybrid alleles. So these are some of the platforms that are available for commercial pharmacogenomic testing. All of them have assays that can detect duplications, deletions. However, not all of them can give you copy count. And to be able to distinguish between these hybrids and the duplications, you need to have copy counts across multiple regions. Some of them cannot detect these tandems and hybrids, and some do. And in this next slide, you can see that a lot of platforms that can detect these CNV, these duplications and these hybrids are actually those that offer copy number variations in multiple regions of the gene, especially the regions in which these hybrids are known, or these conversions to 2D7 are known to exist. Some of them actually, such as Agena, Veridose, or PharmacoScan, have a package solution where the CNVs are fixed and the lab director cannot choose one over the other. Other solutions allow the lab to choose which CNV to run, and that can also pose problems because that requires expertise from the laboratory director to know what they have to order and how to do interpretation. And for us as an interpretation company, it adds another layer of complexity because we need to account for all these different inter-platform variability when we do our translation. So the first case study I want to present, and I'm sorry I'm a little bit behind schedule, is this is just an example of a common platform that a rather small panel, a couple of common SNPs to detect common alleles. And by simply looking at the presence of these two variations, the 2850 and 4180, according to the old nomenclature, we can see that both of these are heterozygous based on the reads that we have in 3 regions actually of the gene. So 3 CNV results are giving us 3 copies. So a simple look at this information would allow a lab to say, well, it's a star 1, star 2 with 3 copies. If you have a star 1 and star 2 with 3 copies, this is an ultra metabolizer because that we score that as 3 functional alleles. We don't know whether that 1 is duplicated or the 2 is duplicated, but it doesn't matter. In both scenarios, this patient will be likely ultra rapid metabolizer. Now, if we take a careful look at this particular sample, and if we run now an additional structure copy number variation upstream, which is in the 5 UTR region or in exon 1, we realize that there is a drop. So there is a lower number of copies, which indicates that there is a structure upstream that is actually probably not a 2D6. So, and that allows us to now refine our diplotype from being a star 1, star 2 duplication to a star 1, star 13, 2. And because 13 does not contribute to function, we're revising this phenotype to a normal metabolizer. So this is an example of mischaracterization that can happen. The prevalence of this is rather low, as I said earlier, but we need to keep in mind is, although the prevalence is low, the consequence is very high, because we would have been wrong regarding 19 medications that are extremely metabolized for 2D6. So you can imagine if this goes in the medical record and if this stays forever for perpetual use, we better do a better job of characterizing these diplotypes. The second case is another platform, which this time doesn't even give you copy count. It just tells you whether there is a deletion or a duplication. And in this particular example, this particular platform is giving you a homozygous call for the variant allele for the 100C2T, which is a decreased function allele, rather common allele, as we saw earlier. Based on just the read of this platform, we would call this a star 10, star 10 intermediate metabolizer based on activity score. Now, if we run this same sample with a different technology that now looks at all these multiple regions of the gene with the copy count, we realize that we have actually no copy in the exon 9 region. We have 2 copies across all regions, and there is an indicator that there is a hybrid. So some of these technologies can also have an additional indicator for in the software to allow you to ensure the call of a hybrid. And that takes us now from an intermediate to a poor metabolizer, because this individual now is a 36-36. Again, the impact here is, although the frequency for some might argue is rather low, the impact is extremely high, because now this time we have missed and mischaracterized the recommendations for 35 medications. So to conclude on this talk, having discordant CNV result is a great way, is highly informative, but complicated to interpret. So this is required some expertise and some understanding of the limitations of the assays and what an assay can detect and cannot detect. Several 10 carriers that you might see, we have to keep in mind, they might have these complex structures. So 36 tandems, 36 singles, multiple copies of 36 tandems, and that can change completely the phenotype assignment. Currently, we have no standard. What is the minimum SNP list that we have to test? What is the minimum set of approaches that we have to do when we do copy number? So Vicky Pratt is leading a group, and we are actually working in collecting and issuing an article the same way we did it for CYP2C19 and CYP2C9 with the help of AMP into standardizing and giving a tiering approach in what is the minimum list of variants that you need to look at versus an expanded list of variants that you might consider. And finally, there are very limited platforms that can detect these deletion, and it's very important to understand the limitation of each platform and what can be reported and cannot be reported. With this, thank you very much, and I'm happy to take any questions, and sorry for being a little bit over time.
If you don't mind, Houda, we might keep the questions till the end just to give Carissa some time. Now, if you did register for the supplementary materials, they are available at the desks, so please see one of our desk staff on your way out and you'll get your memory stick and your supplementary material. So now it's my pleasure to introduce Carissa Wachtler. So Carissa is currently the Chief Technology Officer and Vice President of Research and Development at AccessDx. AccessDx is a high-complexity, CAP-clear certified laboratory based in Houston, Texas, and offers an integrated genomic solution which includes a unique combination of pharmacogenomics testing using the mass array platform, but also therapeutic drug monitoring. So thank you, Carissa. I'll hand over to you.
Hi, everyone. Thank y'all so much for coming. So my name is Carissa Wachtler, and this morning I would like to talk to y'all about CYP2D6 hybrid allele testing, and just from a laboratory perspective, how we approach screening, confirmation, and look at some of the dilemmas and potential solutions for addressing the complexity of hybrid allele and tandem testing. Excuse me. So just a brief overview of what I'd like to talk about. Just a quick introduction about myself, how I got to where I am today, and then we're going to look at how we approach PGX testing at AccessDx, what the clinical significance is of CYP2D6 for us from the lab perspective, why is it so important, why do we pay so much attention to it. After that, just an overview of the methods of detection, what some of the limitations are that come with those different methodologies, and hopefully we'll have enough time I can touch on the Veritas 2D6 panel overview, and then finally wrap up with a quick overview of the feasibility study that we did at Access in tandem with Agena, to compare our platforms and make sure that we do have continuity from one technology platform to another. So a little bit about myself. I used to live in Florida. I was a marine biologist, had the unfortunate circumstance of being in a car wreck. I ended up moving back to Texas, and my mom insisted that I figure out what was going on because I had a numb spot on my knee. That led to some imaging coming back with a very questionable spot, and so I was actually a patient at MD Anderson for about 6 months, and I decided that maybe I could swim with dolphins later and could focus maybe on something that would help people out. So I ended up going to school at MD Anderson, graduated, and started working at a lab called CompanionDx. And it was a relatively unique time to go into the personalized medicine field because PGX was really just starting to get going. And some of you may or may not know, CompanionDx unfortunately did not make it. But from those ashes, if you will, AccessDx arose. And so we've actually just celebrated our 3-year anniversary on November 1st. We're very proud of that. And we've been fortunate enough to have PGX as a foundation for our testing. And excuse me, so as our testing has expanded, we've also decided to expand the platforms in which we were going to offer the testing, just to be able to provide a more robust and comprehensive solution. A little bit about AccessDx. The history of our staff, as I mentioned, some of us worked at CompanionDx prior, but after that, we all still very much believed in the concepts that were coming out of Companion, and so when AccessDx started, we maintained that foundation of running PGX. So we have a very solid background with extensive knowledge and understanding, the importance of the proper characterization of these alleles and variants that come with tandem alleles and hybrid characterization. So just a brief overview of our panel. Some labs can run 30, 40 genes on PGX. Rather than doing that, we wanted to have a more focused panel. We believe that rather than running so many genes, having a focused panel really allows us to have full coverage of the genes, which allows us to better characterize things like tandem and hybrid alleles specifically for CYP2D6. So for our CYP2D6 copy number testing, we cover exon 9, intron 6, and intron 2. The reason for doing that, like I said, is to be able to offer a comprehensive and full and accurate characterization of these tandem and hybrid alleles, because the patient population that we see is very diverse. And knowing that we could see patients with, East Asian ethnicities and West African ethnicities, it's what we as a lab should do to be able to provide an accurate characterization of these rearrangements. So the significance of 2D6, I think, has probably been well established. I'm going to try and make up for some time here, but just at a high level, 2D6 has gone from having 2 forms characterized to now being associated with over 25% of hepatically metabolized drugs. So as Dr. Hachad mentioned, there's an extremely high prevalence in the Asian population, so much so where we actually had a new clinic come on board that was predominantly Asian population, and we were asked if we were sure that our assays were performing properly because we had such a high increase in star 10, star 36 tandem alleles. So the prevalence is there. It's something that needs to be paid attention to, but you also have to be aware that just because you say you run something, you have to make sure that you're running it correctly. Let me try and go through some more. So some methods of detection, the most common that we see is probably TACMAN on qPCR, but coming in a close second that I've at least seen is the MALDI-TOF mass array. It is a single workflow solution. It's something that can make your workflows and make the turnaround time a lot more efficient, reduce human error, and it's manual setup, which can be cumbersome, but it's easily automatable. So there's just a lot of flexibility that comes with it. Next-gen sequencing, it's really, I would say more focused on certain labs that just want to do next-gen sequencing for everything, or labs that maybe have a high volume but also want to run a larger panel. And then finally, there's still labs doing gels with Sanger and fragment analysis, which is fine. It's maybe lacking in the onboarding, but it is still prevalent, so it's just something to be aware of. So some of the dilemmas. From a lab perspective, what we find is the coverage from one panel or from one platform or technology to another, there's a lot of variation, and so that makes it very difficult for labs and for people in a decision-making position to choose what platform, what assays to run. The slide that I have up here, is this the point? Here at the top shows you kind of the coverage for, this is the CYP2D6 gene, and then it lists out some of the different platforms that are available and shows the coverage of those. And, short of doing next-gen sequencing on every single sample, it's going to be hard to really get a comprehensive view of what's going on for CYP2D6 with these tandem alleles. But again, it's something that you need to be made aware of, or to be aware of in the lab setting, to make sure that what you're doing and the assays that you're running and the validations that you're performing are detecting the accurate genotypes. And it's very frustrating, I guess you could say, when you're trying to validate something, but your reference material isn't giving you the calls that you're expecting, and you have no idea why. So being able to really see and get a broad picture of and comprehensive picture of what's going on within the gene it matters what platform you run on and what assays you use. So the second and probably almost equally important dilemma that we see is just the sheer nature of how highly polymorphic CYP2D6 is. I'm not going to go into too much of the genetics about it because Dr. Hachad covered it very well. But because of that, specifically for the STAR10, STAR36 tandem alleles that we see, there are so many labs that maybe don't run the assays that they should, but it's not, intentional. It's just sheer lack of not knowing. So because of the polymorphisms that we see within CYP2D6, seeing a 2-copy exon 9 when really it should be characterized as a tandem STAR10, STAR36, it's something that we're seeing a lot more frequently just because labs are trying to onboard these assays as fast as possible without giving enough thought or putting enough importance on the proper characterization. So what's next? I mean, why do we care? Why does accurate classification matter? I mean, STAR10 is a predicted decreased phenotype. STAR36 is a non-functional. That in and of itself tells you that there's enough difference between the two that you have to be able to differentiate between them. Excuse me. What happens when you have those together? And not only that, but what happens if you have those together and then have it duplicated? It becomes very cumbersome and very, very difficult to try and differentiate, and knowing what you're doing matters. And then Dr. Hachad also touched on this, but from the lab side, it is, required to, do literature reviews, take a look at what other labs are doing, work in a collaborative manner. But labs need better guidance. Where do we start? What resources, are even available? What controls to use for validations? I'll wrap up really quick, sorry. With the feasibility study that we did, so at Access, we were running the Thermo platform. We did a feasibility study with Agena where we sent 10 samples to be run on the Agena platform, and it came back with 100% concordance. And it was amazing to see the continuity from one platform to another, and to have that validity that what we were doing was able to be reproduced. And it was also exciting to see that Agena's panels covered it as well. So it was very exciting to see the growth of the platform. And that is it.
Thank you, Carissa. Would you mind checking if there's another workshop that needs to bump in? All right. Apparently there isn't, although I appreciate that some of you may wish to go to your other workshops. I'm just going to give you a little bit more information on our new Veridose CYP2D6 CNV panel that we're launching today. This is in combination with our Veridose core for genotyping. So we've heard that the mass array system is based on Maldi-Toff mass spectrometry. I'm not going to give you a long lecture on Maldi-Toff mass spectrometry except to tell you that we use it to accurately weigh nucleic acids to determine the variants that are present. You are welcome to come to our booth and any of our staff can give you a well over an hour long lecture on Maldi-Toff mass spectrometry and how it works. But really the power of a Maldi-Toff mass spectrometer is that we can do many, many measurements very, very quickly. We're not detecting a fluorophore, we're detecting the nucleic acid itself down to its molecular weight. And as such, we're able to get highly accurate both detection as well as quantitation of these nucleic acids. Now, it's particularly well suited to pharmacogenomics because the consumable item inside the Maldi-Toff, which we call a spectra chip, is just a surface that we're using to introduce the nucleic acids to the mass spectrometer. It's not specific to your particular test. So any panel, whether it be our Veridose Core or our Veridose CNV or any of our other panels, all go onto the same chip. So it offers you extreme flexibility in what you can run at the same time, in the same run, on the same platform. Now, laboratories doing pharmacogenomics testing at the moment, and we've heard, often have to use a variety of different technologies in order to get the genotypes, the insertions, deletions, the duplications, amplifications, and hybrids. And that's very cumbersome for a laboratory. But here we offer all of that on that single platform in the single run, giving you all the data that you need without the complexity of running multiple different technologies. So the platform itself has very robust performance, accurate results without the requirement to duplicate samples or run duplicates or run technical duplicates on the platform. Comprehensive detection, as I said, for both those pharmacogenomic SNPs, the insertions and deletions, as well as the copy number variants. And it's highly cost effective. Now, a mass spectrometer has a vacuum chamber inside it. It doesn't have a gel, it doesn't have a polymer. A vacuum is generated by electricity. Right? So electricity isn't that expensive to run the mass spectrometer and run a large number of samples over the course of a day. Anywhere from just a few samples all the way up to 4,000 samples a day can be handled by this instrument. Now, when we were developing this new CYP2D6 CNV panel, we wanted to achieve several development goals. Accurate CNV calling, and calling of these complex variations in copy number variation, not just whether it's an amplification or a deletion, but these complex hybrids that we've heard about today. And we wanted it with an efficient laboratory workflow where you could consolidate all of those different markers onto a single platform. So we've heard a lot about the hybrid alleles. I think we've seen this slide over and over again, so I won't harp on the complexity of these hybrids in CYP2D6. Now, the Veridose CYP2D6 CNV panel has 22 assays in a single reaction, and these assays cover 7 different regions of the CYP2D6 gene. The 5' UTR, exon 1, intron 2, 4, 6, 7, and exon 9. So this gives you very accurate analysis of the copies across various regions spanning the entire gene so that you can determine these complex hybrids. Oops, sorry. Going the wrong way. Okay, so this is an example of a sample where we've got on the right-hand side is the graph of all of those assays across the gene, so all 22 assays across those 7 regions, and you can see this isn't a complex hybrid, this is a standard 2 copy across the entire region. Now, the software itself analyzes all of those assays and then reports out the results. So you get your overall copy number. But you also get your hybrid status, and you also get your functional copy number. So functional copy numbering is the number of functional copies. For example, many of those hybrid alleles are not functional. We have genotyped alleles like star4, the carrier that the one that I have, that is also non-functional. So not just copy number, but also that functional copy number is a part of the report. So 2 studies that I'm going to show you that were part of our verification and validation process. In this first study, we looked at 1,120 cell line samples that had been previously characterized and published in the literature on orthogonal platforms. So a variety of different technologies used here, TACMAN, long-range PCR, and other technologies. And we ran these samples on the new Veridose CYP2D6 CNV panel. Now, we had 90% initial concordance with orthogonal methods. Okay, nice but not great. So then we needed to work out what was going on. So in the majority of the discordant samples, the Veridose CNV panel had actually reported a hybrid allele in most of those samples. So we needed to have an adjudicator. We needed a judge. So we called in Andrea Gaelic in order to determine what the true status of those discordant samples were. Now, just as an overall, in these HapMap samples, 13% of the samples were determined to be a hybrid allele, and 70% of those hybrids were previously uncharacterized. The majority were the star 36 and the star 68, with a small number of star 13s. So we sent it out to Andrea Gaelic to run these discordant samples on long-range PCR, and we found that we had 99% concordant with those long-range PCR results. So many of those samples that had been previously haplotyped, for example, the first one here, which was a star 2, star 4, the Agena panel gave star 2, star 4 plus star 68, hence the discordance, and then run on long-range PCR, that was confirmed in over 99% of the samples. Now, HapMap samples are cell lines, they're not the real world, they're a nice tool to start with, but of course the real proof is whether it can be run on clinical samples. So we ran 909 clinical samples. These were small residuals that were left over from testing in one of our partner laboratories, and they were analysed with two methods. We used the TACMAN probes, 3 different probes, Intron 2, Intron 6 and Intron 9, and this new Veridose CYP2D6 CNV panel with its 22 assays across those 7 regions. All right, so initial accuracy of 98.2%, concordance between the TACMAN and the Veridose CNV results. 7% of this population possessed a hybrid allele, so again, high prevalence of hybrid alleles in these populations that we're analysing. Most of the discrepant results were showing a hybrid allele status on the Agena Veridose panel that were not detected by TACMAN. Now, TACMAN only had, the TACMAN probes that we selected to use was all 3, so those 3 probes, but we only had enough sample to do a single replicate. And as you'll know, it's recommended to do 4 replicates with those probes. So there is potential for false results because of only having a single TACMAN replicate, but also because it doesn't have that diverse coverage across the region. We believe that explains the small level of discordance that we observed. All right, so in summary, CYP2D6, CYP2D7 hybrid alleles do occur at high frequency across all populations, as we've heard today. Our new Veridose CYP2D6 CNV panel is a single reaction panel which accurately quantifies CYP2D6 copy number, even in the presence of these complex hybrids. And it's all in a single reaction on a single platform that couples with our Veridose core panel to provide genotyping across a broad range of relevant pharmacogenomics genes. So thank you for those who did stay on. Sorry for running over. Happy to take any questions for any of our presenters.